welcome to Lunch with the Core. Today's speaker is Yuhan, Wang, and Gopika, Gopan, and they're speaking to us about our new MINFLUX system and the work they've done. So welcome.
Thank you, Glenn. Good afternoon, everyone. I'm Yuhan. This is Gopika. We're from Grubula Group. Today, we're going to discuss a new instrument that the core facility have. It's called MINFLUX. And our title is MINFLUX, Tackling the Puzzle of Protein Reaction Diffusion in Living Cells. Okay, as a new instrument, the first question you might want to answer is, so what is MINFLUX? Let me show you a video as an overview of what is MINFLUX. Okay, so this is a video from their official website. I don't know if I should pay them to play the video or they should pay me to promote the video. So from the video, we can see MINFLUX is a very powerful imaging system that we can use for high resolution confocal image as well as single molecule super resolution tracking. And it works in the living cell as well. So when I introduced this system to my friend, the first question I got was, so what does MINFLUX stands for? Why is it called MINFLUX? This is a really good question. Why is it called MINFLUX? We need to answer this question by understanding how MINFLUX work. So I'm going to explain that by comparing MINFLUX with the epifluorescent microscopy. Okay, so epifluorescent microscopy is what we have normally in our lab for imaging. And how it works is you will have a sample that contains either cell or tissue, and this is what you're interested in of sampling, imaging. And you can shine an excitation laser on it because the sample is fluorescent, so you can get emission light out of that. And if there is a molecule on the sample that you're particularly interested in, then this molecule will be captured, the emission of the molecule will be captured by the camera. And what you can see on the camera is going to be a spot because there is diffraction limit. Fortunately, this spot is going to be a 3D Gaussian distribution. So you can always fit the spot intensity into a 3D Gaussian and localize its center point where it should be precisely telling you the localization of the molecule you're interested in looking at. So if this molecule is not fixed in the sample, actually it's moving around, you want to track it, then what you will have on the camera is when the molecule is moved around, the spot is also moved around with the molecule. And the center of the spot is always move around with the molecule. By telling where the center of the spot is moving, you can tell how the molecule is diffusion in your sample. That's how the epifluorescent microscopy works. So you always look at the center of the spot, which has the maximum photon flux. Okay, now let's go to MINFLUX, minimum photon flux. How is it works? Okay, so you have the same kind of sample placed on the platform, and then the same idea, you can shine a excitation laser on it. And if there is a point, there is a molecule that you're particularly interested in, then you will know on your camera the molecule is going to be somewhere on the camera. But we don't know where the molecule is. And how are we going to know that? We really don't know. Okay, so think about if the excitation laser is of a special shape. It's a donut-shaped laser. And if somehow you can move this donut-shaped laser to where the molecule is at and make the molecule localize at the center of the donut-shaped laser, then where the donut-shaped laser is at is where the molecule is at, right? But we can't do that. Okay, a demon can. Think about, there is a demon in this system. The demon is so powerful and so smart, it can always move your donut-shaped laser to where the molecule is at. So the molecule is at the center of the donut-shaped laser and is not being excited because the donut-shaped laser does not have any intensity in the middle. So the demon can be even smarter than that. When the molecule is moving, the demon can always move your laser with the molecule. Okay, so let's look at this. The molecule is moving, the demon is moving, the laser is moving, and by telling how much your laser is moving, you can tell how much your molecule is moving, because the demon makes the laser move with the molecule all the time. So that's why it is called minimum photon flux, because you're always looking at the center of the laser where you have the minimum photon collected. So this system is giving you no emission and no bleaching. So ideally, you won't get any emission light from your sample because it's at the center, and you will not get any bleaching because your sample was not being excited. Okay, so this is the underlying mechanism of your MINFLUX system. That's why it's called minimum photon flux. As long as we have a demon, we will have a very ideal system. Well, but in reality, there is no demon. We have to figure out what we're gonna do to replace the demon. So what we actually have in the system is three things functioning as the function of demon. So we will have a detector that can detect your emission light. We will have a controller that can move around your laser, and we will have a beam deflector that can change the position of the donut-shaped laser. So what you actually have is you will have a laser, you will have a face mask that help you to reshape the laser into a donut shape. And then this is going to be an excitation light, then excite your sample, and you will collect some of the emission light because it's not always centered. The demon is not functioning yet. You will collect the emission light to the detector, and then detector is going to tell the controller, okay, it's not centered yet, please move. And then the controller will shift your beam to where it should be. And that's how it's working as the demon. Okay, so how you gonna actually localize your particle? So think about you have a molecule that you wanna track in the system, and you have a donut-shaped laser that is somewhere on the camera. It's not precisely focused on the molecule yet, but you know it's somewhere. Then you will, on the detector, you will have, because you will have an excitation laser that is of a donut shape, then you will get some emission light out of your sample. This will give you some emission light. But you know it's not the minimum emission light yet because it's not zero yet. Then what you need to do is to move the donut-shaped laser to where it can give you a zero photon. So what you do is you move the donut-shaped laser, and together with it, the excitation intensity is moving with it, and the photon that you're collecting is also moving with it. So you will see a minimum position here. This is where your molecule is at. When your donut-shaped laser is on that, you're collecting the minimum photon, and you're knowing this is the position of your molecule. Okay, so actually what we need to do is we don't need to scan the full curve, because we can approximately consider this curve as a quadratic curve. So all you need to do is to take 2 points from the start to the end, and by knowing the photon of those 2 points, you can fit this curve in a quadratic function and get the lowest point. That's going to be the position of your spot. So this is how you use MINFLUX to determine where your molecule is at. The position is measured and determined via the minimum photon flux. So this is a simple example of moving your donut-shaped laser in 1D. In 1D, you only need 2 points to localize your molecule. Let's move on to a little bit complex system in 2D and 3D. So in 2D, what you will need to determine the position of your molecule is not only 2 points, but at least 3 points, better to be 4 points. So the same idea, your donut-shaped laser is going to scan through this point 1, point 2, point 3, and finally localize to the point 4 to know where your molecule is at. This is the first scanning. You're scanning in a big range to kind of localize, like, vaguely the position. And then you can always shrink the scanning area to make the scanning area smaller, and then you can localize it more precise, and then scan it even smaller to localize it very precisely. This is the 2D. 2D need 4 points. And for 3D, you will need 4 points on the 2D plane, and then 1 on the top, 1 on the bottom. So 6 points in total. The same idea, you are first scan it in a broader area, and then you shrink the scanning area more and more closer and identify the localization very precisely in the end. So this system is giving us less bleaching, because in ideal case, there will be no bleaching. You're always collecting the minimum photon position, so very less bleaching, and it gives you very high resolution because you're scanning very fast. You don't need to wait for photon to be collected for a long time. You're doing minimal, so it's going to be very fast. And of the high resolution, because your resolution is also going to be determined by how closely your scanning range is. So less bleaching, higher resolution, faster checking. That's the advantage of using MINFLUX. Now the next question is, okay, good. We have a really powerful imaging system and tracking system now. What can we use for study? What application it can have? Okay, so let's discuss our project. Our group, the group, is focusing on studying protein dynamics in the living cell. We know in one human cell, there are more than 20,000 proteins. And those proteins perform the majority of their functioning inside the cell. However, the cell is not as simple as a test tube. The cell contains water molecules, ions, DNA, RNA, other protein molecules, a lot of things. All those molecules are forming a really complex matrix of interaction network. So the protein is performing their function in this interaction matrix. So when we study protein dynamic, protein behavior, it becomes extremely important to put the protein in their native environment, not only the test tube, but in the living cells. So our group has studied protein folding in the living cell, protein RNA binding in the living cell, protein-protein assembly in the living cell. And all these studies are done on the enzyme level. So it will be extremely cool if we can study these protein behavior in a single molecule level. And MINFLUX makes it come true. So let's think about this system in a more schematic way. We have several proteins that can diffuse around in the cell. However, they're not just having in the solution, we have so many other molecules that are blocking their ways. So the protein is diffusing and also have interaction with these molecules. So how we want to know the protein's diffusion and interaction at the same time, then what we can do is we can pick one single molecule and get its trajectory. By the trajectory, we can tell how much the protein is diffusion and how much the protein is interacting. And we can fit the trajectory into a curve, and by using MINFLUX, we're able to get that kind of trajectory. Okay, let's look at the system in a more close way. Think about this is a protein you're interested in looking at, and this is a molecule that the protein is going to bind to. So when those two molecules are far apart from each other, then they're not binding to each other. They diffuse fast. Once they see each other and they start binding, they will diffuse slowly together. And then it's an equilibrium, so they will be apart again, and they will diffuse fast again. So by measuring how fast they're diffusing, we can actually tell if they're bind or unbind. So in the cell, in this interaction matrix, the protein is diffused around and bind more particles in this complex matrix. And this reaction, this binding reaction, is also affecting protein's diffusion and tumbling. So it will be good if we can study the diffusion and interaction at once in the living cell, because they're not two independent behavior. They're the behavior that coupled to each other. Okay, so how are we gonna study protein diffusion and interaction in the living cell? We first picked a model system. The model system we pick is a protein RNA complex. So the protein is U1A, the RNA is its binding partner, SL2. But this is a standard protein RNA binding system. That's why we picked it as our model system. It has been well studied in vitro and in human cells. So to study their diffusion and interaction in living cell, we break down our project into 3 major steps. First, because MINFLUX is a new system, and this is our first time to study MINFLUX in a live cell tracking, so we first need to select the best dye that works for labeling and tracking of our system. The second step is we want to see whether we can actually tracking single molecule in the living cell with MINFLUX. And third, we will come to our model system. It can be this model system, protein RNA complex, or it can be another model system that we'll discuss later. It's a protein-protein complex. Okay, so let's do this one by one. First, selecting the best dye for labeling and MINFLUX live cell tracking. So we have a list of candidates that we tried one after the other. And how we do this is we have the dye that hook up to some molecules, and thank Selvin Group for offering all those dyes. What we do is we pre-mix the dye with our tracking buffer, injection buffer, and then we use a microinjector to microinject this mixture into individual live human cells. And the cell is still alive after the injection, but the cell takes the dye. And then we bring the sample to MINFLUX for a live cell tracking. So what we can get is, let me show you some example. what we can get is we can get a cell image. On the left channel, we can see the cell shape because the cell is co-microinjected with a green dye that is called Alexa-488. So we can see on the green channel the cell shape clearly. And we can see a nucleus in the middle and a cytoplasm. And on the right-hand side, it is a channel that is for the red. So in this channel, you can also see a cell shape. That means the cell is actually containing the red dye that we are interested in testing. So we can see a cell shape. That means our red dye is located in the cell about uniformly. Okay, so let's do a live cell tracking and see how the traces look like. Ooh, we got really tiny, small, short traces. That's not good. That's because the auto 647 is known for being very sticky in the cell. So in the cell, it's not performing the best functioning as we would expect. So this is a bad dye example, auto 647. Let's look at something else. On the left, it's the same idea. It's a green channel that gives you the shape of the cell and the nuclear. On the right-hand side, it's another dye that's called Auto 655. It gives you a shape of the cell, and it's located uniformly, and you're not getting a lot of long traces as well. So this is another bad example, Auto 655. It's also because the auto is sticky, so you don't have a lot of free-moving molecules in the cell. And a third example is Alexa 647. So on the left, you can see the shape of the cell. They're stick to each other, but you can clearly see the boundary. But on the right-hand side, it's very dim. That's because, and for the tracking, you're not getting a long trace. That's because Alexa 647 is not very photostable, so it's also not the ideal dye for tracking. It's another bad example. Okay, so what can be the good example? Now we know we need something that is not too sticky in the human cell and also has a higher photostability. Okay, whatever. Okay, so let's look at this dye. This is CF633 that hooked up to a streptavidin. And on the left, you will see a clear cell shape that give you a boundary of the cell and a nuclear. And on the right-hand side, you can see the streptavidin with the dye is located mostly in the cytoplasm because you have a dimmer area in the nuclear area. Okay, so if we perform a MINFLUX tracking, first we select the closer area, like around here, to do the MINFLUX. We specifically select the area that has half inside the cell and half outside the cell. So once we get the tracking traces, we can tell if it's from the molecule inside the cell or just from the background. Okay, and then we did a tracking. Boom, this is the long traces that we have, and all the trace we can see it's located in the cell. We can see this clear boundary. Good, so this is a good dye example, CF633. Please remember that. Okay, and another one that we also tested is called LD655 melamide, and we microinjected into the cell, crop this area for MINFLUX tracking. It's also half within the cell and half outside of the cell, and boom, we're seeing those traces. Those are long and located within the cell only. Okay, so this is another good example of the dye, LD655. So far we know CF633, LD655 are the two dyes that we would use for MINFLUX live cell tracking. The property of those dyes are they are very photostable and they don't stick a lot in the live human cell. Good. Now we have our best dye selected. The next step is to actually track the single molecule. Let's look at some quantitative data. This is the same cell that we are tracked with streptavidin hooked up to CF633 dye. And we're getting these traces on MINFLUX. And we can replot this trace on the MATLAB, and the trace looks like this. each individual trace were plotted in a different color. And actually, we're looking at this in a 2-dimension. Actually, this is going to be a 3-dimension. And we can zoom in to each individual trace. It will look like this. So this is a trajectory that one molecule diffuses around for about 80 milliseconds. And this is another example of another molecule that diffuses for longer time, that's more than 600 milliseconds. And from this trajectory, we're able to plot the mean square displacement over time and fit the curve into a linear function. It's quite linear. And from this curve, we're able to tell for individual molecules what is the diffusion coefficient. So let's take a look at the diffusion coefficient distribution. So comparing the in vitro to the in cell, we're seeing a sharp peak on in vitro and a broader peak of in cell. That's because the cell is a complex system, so the molecule will have a broader diffusion coefficient distribution. And we can also look at individual molecules. So we can select a molecule from the slowest side, the middle side, and the fastest side. So this is the fastest side, slowest side, and the middle side. Wait. And we can plot their mean square displacement over time. And from this kind of curve, we're able to tell whether they are subdiffusion, normal diffusion, or super diffusion. So those are the data that we can collect and analyze from the MINFLUX system. Now we have our best dye selected. We have a single molecule that can be tracked in the live human cell. Let's move on to next step. That's going to be our actual system. We have 2 model systems that we are particularly interested in studying. One is a protein RNA complex that I already described before. One is a protein-protein complex that comes from a virus capsid system. So I'm going to end my part with a photo, and then I'm going to introduce my lab mate, Gopika, to continue with those 2 actual systems. So I'm going to end with this happy photo when the MINFLUX just arrived. We took this photo. Everyone was smiling so bigly. So, okay, so let's move on to the next part, the actual system of protein RNA complex and protein-protein complex. Welcome, Gopika.
Thanks, Yuhan. Hi, everyone. So I'll take it over from here. So yeah, as she was describing, so I'll introduce you to the 2 higher systems or complexes of proteins that we're eventually interested in looking at using MINFLUX. And the first one is a protein RNA complex. So here, the system that we're looking at is, again, U1A protein and its binding partner stem loop 2 of the U1 RNA. So this is a part of a bigger complex called the spliceosome, which takes care of the splicing in eukaryotes. And it's a very well-studied system, and it's a model system for the protein RNA interaction. And our group has looked at the binding of this complex inside live cells. And we have characterized the binding constant to be around, the dissociation constant to be around in the micromolar range, which makes the system suitable for our applications and what we are planning to look at using MINFLUX. So again, the question is, how is the protein RNA binding and diffusion coupled inside the living cell? And just to remind you, so what we are hoping to see and study inside the cell is that since we have the binding equilibrium of the protein and the RNA, we assume that when they're individually diffusing, they diffuse faster, they bind to each other, diffuse slowly, and they eventually unbind and again diffuse faster. So in this case, our plan is to label the RNA with the dye that we have already selected. The dyes that we have shortlisted, basically. and look at and inject the protein with the labeled RNA, co-inject them into the live cells, and track the diffusion and binding in 3D. So we're just holding up so we have everything ready right now in terms of the sample, and we're just holding up the measurements for the 3D mirror that's getting fixed in MINFLUX. And moving on to the second system, it's again a larger complex of proteins. So it's a hepatitis B viral capsid, which is formed from 120 homodimers of the capsid protein, and it's 35 nanometers in diameter. And why do we want to study the viral capsid? We already have projects running in the lab where we're interested in answering certain questions that are still debated upon and in the field. So hepatitis B virus, in spite of having a vaccine that works, it still kills close to 1 million people every year. And this is the prevalence of hepatitis B provided by CDC around the globe. And so we think that we, with our instruments, both in the lab and using MINFLUX, we'll be able to answer certain questions that are crucial in terms of how the virus actually attacks a cell and how the life cycle happens inside the host cell. So this is a very schematic summary of a typical life cycle of hepatitis B virus. So the virus enters the cell, and it's important to the nucleus for DNA repair and replication and further infection. So the part that we're interested is mostly here, which is the most vague in terms of the mechanism that people know in the field currently. So that there's still the unanswered question at how the capsid actually disassembles inside the cell, where it disassembles to actually release the genome into the nucleus, which is the actual goal of the virus after entering the cell. So what we have is a viral capsid with M-cherry protein, which is a fluorescent protein. So each of the monomer is labeled with an M-cherry, and so the capsid has 120 M-cherry decorated on the capsid. And this is just a schematic again, just showing the M-cherry at the boundary. And we express the capsid labeled with M-cherry in the in hepatocytes, liver cancer cells, HepG2. And here you see the confocal image from MINFLUX. On the left side, it's a bright field just to see the boundary of the cell. So unlike the previous images we saw, we're expressing the protein in the cell, so we're not able to inject any other dye to actually visualize the cell, so we use a bright field. And we observe that the cells are pretty crowded, and this is, again, the preliminary image or measurement that we could manage to get with the 3D mirror working. And so even at the right side, you can see the dark regions that you see are the nucleus. So those are the cells, and it's pretty crowded. But still, the red regions that you see is actually the capsid protein labeled with M-cherry. And so we selected this region, and we did a tracking, and we could get some really good trajectories of the protein. So here's a zoomed in image of a representative trajectory. It's gone around 1.5 seconds. And following the same pathway that Yuhan described earlier, so we can get the diffusion coefficients of the capsid, and we see that there's a range of diffusion coefficients inside the cell, which is expected because the cellular environment is, again, very diverse. And we can look at the different ranges of diffusion and characterize them into subdiffusion or superdiffusion. And again, we're waiting for further measurements in 3D. And one of the other questions that we want to answer is, again, how the actual capsid diffusion happens and how it happens around the nuclear membrane. And so this is like just an example of how different hypotheses on how the capsid disassembles at the nucleus or the nuclear pore complex, and we hope that we'll be able to see something using MINFLUX, which will help us answer the question. And we already have expressed the capsid with mCherry in the FG2 cells, and we can label an nucleopore complex by immunostaining. And look at the system using MINFLUX for diffusion. Again, I would like to thank Martin, our advisor, and Zan for their insightful suggestions and help throughout the project. And Glenn and Kingsley, again, for helping us on a daily basis for troubleshooting at every step and training us on the instrument. And we'd also like to thank Selvin Group, Kim Group, and Golding Group for providing all the dyes that we used for staining in the initial part of the project. And yeah, that's again the happy picture we had when the instrument was being installed. Thank you.
Are there questions after that?
I think Mika has a question.
Technical question. Maybe should I wait? I have two questions. The first one is the phase mask, what shape does it need?
The face mask, it's like this shape.
The shape it makes the beam and maintains it as it moves?
The face mask is how it creates the donut-shaped laser. Both in 2D, it can be done in 2D donut shape or 3D donut shape.
How do you make 3D donuts?
3D donut shape, you will have something on the top. It's actually like a hat. You will have something on the top, and then a free space in the middle, and then something on the bottom. So like basically you have a free room that in the middle that can capture the molecule. I should have a slide. Hold on.
Have it after this, right?
Yep.
I mean, like, does it move to the top? Is the donut?
Oh, it's hidden, I think.
It should be like a sphere with the core removed.
Yep.
Not
How
like a-
can
not
I
like a-
see the hidden
Can
slide?
Not like a toroid, but
slideshow.
a sphere with
The
the.
intensities on the top and bottom are higher, and then there's a donut in the shape.
There we go.
And the way you make that is by using a top hat form for the phase space. So there's a spatial light modulator that changes the phase, or makes the phase mask. You want to set that up for different wavelengths, right? So if you use your top hat, you want to too low.
Yep. So this is the image that from the, for 1D. That's the first one is a standard. It's not the donut shape. The second one is how you create a donut shape in 2D, how it looks like from the side, how it looks like from the top. This is how the 3D donut shape look like. So if you cut from the middle, it's also gonna look like a donut. But if you look from the side, it has a hat on the top and had a hat on the bottom, and has a space in the middle.
Cool. Thank you. And then the second one is like, so there is bright fields in the.
Yeah, there are bright fields. So before, what we are testing with our live cell sample is we co-microinject 2 dyes, not only the dye that for MINFLUX tracking that's red, we also co-microinject the green dye just to see the cell shape on the eyepiece. Otherwise, you don't know which cell has the molecule, which cell does not have the red molecule. Because the red molecule is very, very dim. It's like nanomolar range. You will not be able to see it from the eyepiece. That's why we co-microinject the green one. But it comes out, the green will give you background. So it will be ideal, and we're going to do that in the future, is we don't microinject green at all. We just have the red, and we look at the bright field to see where the cell is located. And then we just image the red channel. So you will have 2 channels still, but one is bright field, one is red channel.
I
Yeah.
guess I'm
Don't
asking
use the green.
When we did it, we were told we could only have reflected light, and it just gave like a really big boundary to ourselves.
No, you can do bright field imaging.
Was on
I don't even know why they said that. It
there was no
it
bright
has a
field?
bright field.
It works. It works very well. The bright field looks very well.
Okay, that's good.
There might have been I don't know
Is it
if
new?
there's
Like,
something with your sample, I don't know.
oh, maybe, is it, what are like, what do you need for it to work? are you saying that like we didn't, no, you told us we didn't have that field, so we did reflected light instead?
No, then that bubble was in this one.
Yeah.
We have that. So
Yeah, you have, we
You have
have,
bright field.
they have bright field. We have, you have.
And there's no reason that bright field would raise photons or anything, right?
Nope, nope.
So,
It's independent.
bright field.
We're just using that to find your sample before you start. So that's.
That's, yeah.
Any other questions? Yes.
So, excellent talk, by the way.
Thank you.
I'm most interested in the bad dye example.
Bad dye. Are you using bad dye? Are you the one of the victim?
So I do know a lot of tracer, they're sticky. Do you know, do they stick to the basement, the membrane itself of the cell, or like the cytoplasm or any like albumin?
We don't use this dye other than MINFLUX, so we can't give you like a very accurate answer, like whether we didn't test, because once we know that it doesn't work, we just trash them, right? So, yeah, I think Gloria might have a better experience because she worked with the dye. Yeah, stick to anything that's charged.
Oh, that's what they do to charge for the
Yeah,
tracer.
so normally
Yep.
if you want to use ADO, you use the ADO situation where you have fixed stabilized cells that you can block the strikes.
Mm. Okay, so that's why you don't use ADO for tracking. Yep. Yep.
How do you characterize subdiffusion and superdiffusion?
Oh yeah, this is very preliminary data. So we will collect more new data once we got the 3D mirror again. But the subdiffusion and superdiffusion, you can tell from the trace. One is gonna be curved, one is gonna be like go higher, right? So from the trace, you're able to tell whether it's leveling off, whether it's like always linear, or it curved off, or like it goes higher.
So is it like the value of the diffusion coefficient based on which you're like categorizing them?
Yeah, so you plot the mean square displacement over time, and then to see if you can fit it into a linear curve very well, or it's more of going up as a linear curve, or it's more like goes to saturate as a linear curve. That's how you like identify them.
Yeah, it's that plot in the inset that we're talking about. So
This one. This one. So if it's like goes up, then the linear curve, then it's a super diffusion. If it's a linear, then it's a linear. If it's like leveling off, then it's yeah, the slope. The linear is like not changing the slope over time, with a different time lag. It's not changing, but the super and the sub is gonna change. Yep.
You can only measure one particle at a time, correct?
Yeah. Are we Kingsley? We're checking. We're checking more. Multiple at a time. Yep. So you can see like those traces. They're not being captured at one after the other. Like for multiple molecules, they got trapped at the same time, tracked at the same time, and then move on to the other side.
So then do you see either quality changes or length of tracking changes depending on if the molecule was measured earlier in the experiment or later?
Could you repeat your question? Can I repeat your question because I'm not hearing it clearly? So you're asking whether the result's going to change if I track it earlier or track it later.
Yeah.
Well, I don't know, because you cannot track the same molecule again. You don't know, right? If the molecule is being tracked again or not. But we can always do a average and see if the early time tracking versus the later time tracking, if the molecules are like perform differently or not. We can do a statistical answer, but not for one specific molecule.
Yeah, I was just curious.
Yeah.
If the quality was better earlier before you exposed your sample to the donor moving around versus later in the experiment.
That's a good question, but I would still think the bleaching of the MINFLUX is very mild compared to other microscope I've ever used. So I'm not worried about the bleaching effect too much on the MINFLUX system, and the sample stays very bright in like throughout the whole image. So, yep.
So 2 questions, I guess. Following off of that one, if you're able to track multiple molecules at one time, does it imply that you have multiple beams, or is the same beam being used?
No, that I think was that was a confusion. You can only track one molecule at a time.
Oh, okay.
You track them one after the other.
Okay, they were tracked one after the other. Yeah.
Okay.
The same molecule will be tracked at different times,
Yes.
but it's sequential.
Okay.
Oh yeah. Yeah, I was also thinking like if there are multiple beams. Got it.
Okay.
Yep,
And then
that makes sense.
in order to track these molecules, the fluorophores need to be sparsely distributed, right? So that you can localize a single fluorophore at a time.
The labeling is very important.
How do you make sure in these experiments that the fluorophores are distributed sparsely?
In our experiment, we are micro-injected the streptavidin that is labeled by the fluorophore. So like we already know it's a pre-made sample that we already know the protein is labeled by single molecule.
Okay.
So it's not a problem in our case. But if you're labeling like the sample, you're labeling the sample by yourself to do a confocal image, then the labeling might be tricky. The concentration you're using, how much you're incubating, is gonna vary.
Okay, cool, thank you.
Yep, Charles.
Does it switch between molecules because it loses track of them, or is it programmed to watch for a certain amount of time and then look for something else? You just get to sample more molecules.
To sample more molecules. There is a software that we use for the tracking. What was the question again? I didn't quite.
So if it's track, can it keep track of a molecule basically indefinitely? Do you have to program it to switch to
It will switch to other molecules after a while because the molecule is not stable all the time. So once it loses tracking, it will jump to another molecule and track.
it loses track of them?
Uh,
So it can lose track of them, but you also sometimes program it like an upper limit of how long it takes to catch it. So that way it switches to a new molecule, or you just watch one molecule until it loses it.
yeah, watch one molecule until they lose it, and then they go to the other one. So for some of the trajectory, we are getting really long, like 600 millisecond. For some, they lose track very fast, 70 millisecond. So it varies from trajectory to trajectory.
The molecule undergoes that rhythm and then comes back again. So very likely leave sure they get stuck in the state and then experiment from that. I don't think it's wrong.
Okay, dokie.
The question, I wanted to comment on your question. So they went through and did all the things they said about making sure the fluorophores are low concentration, right? And that's absolutely right. There's other things you can do. So when you do the search pattern, there's a lot of little ratios you can check to see if you have two fluorophores, then the ratios between the center and the outside points are not the same as if there's one fluorophore. So you can do those kind of ratios. There's a whole list of those kind of things that you can check to verify that each track is a single.
Oh, okay.
So what they said was correct, and that absolutely works. But there's additional ways that MINFLUX has to double check to make sure you're tracking them.
to make sure that the fluorophore that you're watching is consistent with the fluorophore that you expect you're watching, for example. Okay. Okay, cool. So that helps, I guess, sort of make sure that you're consistently following one molecule, even if there's not quite the sparsity that you expect.
So they record all the number of photons that you collect at each search point, and from looking at all of that information, you're getting a lot more information out than single molecule localization like phosphine, for example. Because there you're just getting number of photons at a location. Here you've got these search patterns and they do the multiple steps. So to one location you've got 12 or 14 different photon count numbers in different locations.
Okay. Okay,
detectors, and you can make sure that the ratios between the two detectors are constant.
Okay. Cool, thank you.
No, it's so you can do two color. It would be nice to do two color images with MINFLUX, right? So it's difficult if you have to set up two channels with this because you don't know which molecule you're tracking when you start. So at the end, you put a dichroic in and you split your beam and you add them together when you're doing the tracking, and then at the end, you look at the ratios of those two after you've done the localization, and that will tell you which color it is. So that's how you can do two color tracking. Are there other questions?
Okay. Thank you, Glenn. Thank you, everyone.
Yeah, so if you guys have further questions, talk to one of the core staff, me and Kingsley or Austin, and we. That was very good.
Thank you.
That was great. Do we like shake hands?
Demo, that's my first time. When I prepared the slides, that's my first time
That was
knowing what is demo.
That was the first
We
thing that-
always like